Students, in today's lecture we have to study about the transportation across the cell membrane. We have studied that cell membrane is a semi-permeable membrane. For the normal functioning of the cell, it is very necessary that the transportation of material can take place across the cell. So let us study about it. So today's topic is transport across plasma membrane. Transportation of substances across plasma membrane of cell membrane is very necessary to maintain the normal functioning of the cell. Lipids, as you know that cell membrane is made up of lipid. Only those molecules which are soluble in lipids or those molecules which are very small in size can pass easily through the lipid bilayer of the cell membrane. But those molecules which are insoluble in lipid and those molecules which are charged are polar molecules such as sodium ion, glucose, uh, chloride ion. They are impermeable because they are polar and lipid bilayer is hydrophobic in nature, so it doesn't allow these particles to transport across the cell membrane. So how they get into the cytosol of the cell or outside the cell? These substances passes through a specialized protein channels. There are some channels, as I told you when we studied about the plasma structure of plasma membrane. There are channels, carrier protein, and some active pump mechanism that help in the transportation of these polar molecule and charged molecule across the plasma membrane. And still there are large molecules which cannot transport across the cell membrane easily. So how they get into the cell or outside the cell? There are for example cell or solid particles, viruses. How they can get into the cell? They get into the cell or out of the cell by the specialized transport system that we studied in upcoming slides. We call it as vesicle transport system. So let us study the methods of transportation across plasma membrane. If we classify the transportation of transportation across plasma membrane, we can subdivide them into three subcategories. That is passive transport, active transport, and vesicular transport. Passive transport, in which the transportation of material across the plasma membrane takes place due to the concentration gradient, that is from higher concentration to the lower concentration. While as in case of active transport, the transportation of material takes place against the concentration gradient, and so it requires energy. And in case of vesicular transport, the large molecules like cell, like bacteria, solid particles, they get entry into the cell or exit from the cell by the vesicular transport system. We will study about it in upcoming slide in detail. So if we want to in further, passive transportation can be divided into four subcategories. That is, four subcategories like simple diffusion, in which the transportation of material take place due to the concentration gradient, and osmosis, filtration, and dialysis. Simple diffusion further can be divided into two subcategories like simple diffusion. In simple diffusion, the fat soluble or lipid soluble molecules or simple small molecules get entry or exit from the lipid bilayer due to the concentration gradient. And in case of second is facilitated diffusion. Facilitated means there are other molecules like polar molecules, and charged ion which cannot cross the lipid bilayer. So how they can get the entry or exit from the cell? They get the entry out of the cell, or they get entry and exit from the cell due to the presence of channel protein in the plasma membrane, as we have studied in detail when we have studied the plasma membrane. This channel protein provide a hydrophilic environment and form a pore from which these molecules can get the entry or exit, there are two types of channels, that is open or gated channel. In open channel, these are the channels which are open throughout the time, and the transportation of material take place due to the concentration gradient, and when there is the equilibrium exists, the transportation stop. But in case of gated channel, they are closed but only open when there is a signal from in the form of ligand, or in case of ligand gated channel, or in case of voltage gated channel, where the electrical signal decide the opening or closing of the channel. Gated channel can be divided into ligand gated and voltage gated. And next is a active transport system, which I have told you that it is against the concentration gradient and require energy. It can be divided into primary active transport port, where the direct ATP utilization help in the transportation of material across the plasma membrane, and other type is secondary active transport, where the directly energy is not used directly, but it create the concentration gradient, which allow the secondary active transport of the material. So, and third type of transport is vesicular transport. It can be divided into two forms, whether the particle is entering or exiting the cell. When it is entering the cell, it is known as endocytosis. When it is exiting the cell, it is known as exocytosis. So let us study the diffusion in detail. As I already told you that it is a passive transport system in which a molecule moves from an area of higher concentration to the area of lower concentration. Material move within the cell cytosol by diffusion, and certain material move through the plasma membrane by the diffusion. Diffusion expand no energy. Energy is not required in the diffusion. Here, if you look at the concentration of material, here is a higher concentration and there is no molecule, so the concentration gradient is devolved, and the molecules from higher concentration to lower concentration take place. If you focus here, that due to concentration gradient, the movement takes place, and it takes place until when there is a equilibrium is maintained. When there is equilibrium, there is no transportation of material take place. So diffusion take place mainly because there is a concentration gradient. When there is no concentration gradient, diffusion will not take place. So what are the factors that affect the process of diffusion? The first factor is concentration gradient. What is concentration gradient? It is the difference in concentration of a molecule across the plasma membrane. When there is a concentration gradient, the molecules transported from higher concentration, diffuse from higher concentration to the lower concentration, and diffusion is directly proportional to the concentration gradient. Higher the concentration gradient, higher is the diffusion, and vice versa. The second factor that affects the diffusion is mass of the molecule diffusing. Higher the mass of the molecule, lower is the diffusion, and lower the mass of the molecule, higher is the diffusion. Third is temperature. Temperature increases the kinetic energy of the molecule, so it increases the diffusion. So temperature is directly proportional to the rate of diffusion, while as lower temperature decreases the rate of diffusion. Solubility. Solubility is another factor that affects the diffusion. Higher the solubility of a molecule in hydrophobic lipid, more is the diffusion, and polar molecules cannot easily diffuse because they cannot cross through the lipid bilayer. Other is solvent density. Higher the density of the solvent, lower is the diffusion, because denser particles move very slowly. So diffusion rate is inversely proportional to the solvent density. Another factor that affect the diffusion is surface area and thickness of the plasma membrane. Surface area is the area, higher the surface area, higher is the diffusion rate, while as thickness of the plasma membrane decreases the diffusion rate. Distance traveled via molecule is directly inversely proportional to the diffusion rate. If a molecule has to travel a large distance, it diffuses very slowly, and the diffusion rate is inversely proportional to the distance traveled. Sometimes the rate of diffusion is enhanced by causing the substance to filter more rapidly by applying pressure. This happens in case of kidney, where blood pressure forces large amount of water and accompanying dissolved substances solute out of the blood and into the renal tubules. The next important topic is facilitated transport. As I already told you that facilitated diffusion is a process by which molecules are transported across plasma membrane with the help of membrane protein. A concentration gradient exists that would allow ion and polar molecule to diffuse into the cell, but this material repelled by the hydrophobic part of the cell membrane, so they cannot cross the cell membrane. So how they get into the cell membrane? The channel protein come in the rescue of these molecules. They aid in the facilitated diffusion of substances by forming a hydrophobic passage through the plasma membrane, through which polar and charged substance can pass. So the channel protein which help in the facilitated diffusion are the special type of protein. They are integral protein which has a special arrangement of amino acids in the cell membrane, providing a hydrophilic passage for the water and small polar ions. Aquaporin is an example that allow water to pass through the membrane at very high rate. Type of the channel protein. There are two types of channel protein, that is non-gated and gated channel proteins. Non-gated channel protein open all the time, while as the gated channel protein remain closed until it get the signal in the form of chemical or in electrical form. So if you look at the non-gated channel, they remain open, and the diffusion take place. Diffusion takes place due to the concentration gradient, and it takes place until there is a maintenance of a equilibrium is attained. Until then, the molecules can move across the membrane. But in case of gated channel, a signal is required so that the gate is open. When a signal is attached to the protein, a conformational change take place, and the gate is open, and the molecule can easily enter or exit from this gate. And this also take place due to the concentration gradient, and the moment until this molecule is attached to this protein, they remain open, while as when it gets separated, the diffusion stops because this channel get closed. There are some examples of these channels. In some tissues, sodium and chloride ion passes freely through open channel, whereas in some other tissue, a gate must be open to allow passage. In kidney, both type of channel are present in different part of the renal tubule, and cell involved in the transmission of electrical pulses like nerve cells and muscle cells have gated channel for sodium, potassium, calcium in their membrane. Next important proteins that are required by the cell for facilitated diffusion are carrier proteins. These are proteins that carry substances from one side of the biological membrane to other. Carrier proteins are typically specific for a single substance. Uh, that's why the glucose carrying proteins can carry only transport only glucose. They cannot transport the nucleotide or sodium or another molecule. Example of this process occur in kidney. Glucose, water, salt, iron and amino acid needed by the cell are filtered in one part of the kidney. If you look at the structure of carrier protein, these are the integral protein, membrane protein, and they take the molecule from outer side and transfer them to inner side. The binding of these molecule to the protein bring the conformational changes to the protein, and it transport the protein from outer side to inner side, and those carrier protein are also there. Those can transfer the molecule from inner side to outer side. The next important passive transport method is osmosis. Osmosis is a movement of water or solvent across a membrane from an area of low solute concentration to an area of high solute concentration. It occurs when there is a concentration gradient of a solute within a solution, but the membrane does not allow diffusion of the solute. This happens in case of polar molecules and ions, etc. Semi-permeable membranes, also termed as selectively permeable membrane or partially permeable membrane, only certain molecules or ions to pass through by the diffusion. If you look at the mechanism of osmosis, concentrate here that here you have only few molecules of a solute, and here there are so many molecules, so here the concentration is low and low concentration, and in other side there is a high concentration. What this plasma membrane or semi-permeable membrane does not allow these molecules from this side to this side, so that a equilibrium is maintained. To maintain the equilibrium, what happens is that this water molecule move from here to here, so that the equilibrium is maintained, and you can find that the water has moved from here to here from here to here to maintain the equilibrium. So when this type of diffusion take place, this is known as osmosis. Next important term is tonicity, which is directly related with osmolarity. Osmolarity determines the direction of water flow. Osmolarity describes the total solute concentration of a solution. Solution with higher solute concentration has low osmolarity, while has high solute concentration has high osmolarity. Depending upon the type of osmolarity, we have three types of tonicity. We have uh, hypertonic, isotonic and hypotonic uh, osmolarity. In hypotonic condition, we have high osmotic pressure than other. If a cell is placed in a hypotonic condition, that is when the concentration of solute is higher inside the cell than the outside cell, the water will enter from outside to the inside cell, which lead to the swelling of the cell. And if uh, there is more water enter into the cell, this will lead to the this will burst the cell will burst eventually. And the next type of tonicity is hypertonic. It is reverse of hypotonic, where the concentration of outer medium is higher uh, than the environment inside the cell. So what happens? Water from here, from the cytosol, leave to the outer spaces, outer environment, and cause the cell, and it causes the cell to shrink. And third type of osmolarity is isotonic, in which the solute concentration is similar inside and outside the cell, so the water can enter and leave and maintain the equilibrium. So with this we conclude our passive transport system. In next video we will study about active transport system. Till then, stay safe, stay at home. Thank you for watching. See you in next video.